So if we're talking about cell division, at some point the cell, well, should actually divide. But so far, all mitosis has done is produce a cell with two nuclei. Cytokinesis is the process in which the cytoplasmic content of the cell is divided into two new daughter cells. Now, while it's easiest to consider the events of cytokinesis separately from mitosis, in actuality, there is some overlap between the two. Lucky for us, the mechanisms that govern cytokinesis are far less complicated than those of mitosis. Mitosis was all about ending up with a precise number of chromosomes. On the other hand, division of the rest of the contents of the cell is far less precise. Having an unequal number of chromosomes in the resulting daughter cells would pretty much be a bad thing. But having an unequal number of, say, ribosomes or mitochondria isn't. It's because the total number of organelles, proteins, and other molecules floating in the cytoplasm is so big that even an arbitrary division of the cytoplasmic content gives each daughter cell a sufficient amount of functionality to survive. So for instance, if a dividing human cell had, say, 1,000 mitochondria, a division of 450 in one cell and 550 in another cell still means that both of the cells can produce enough energy, even though one daughter cell has 10% less energy producing capacity than its mother had. So how do we turn one cell into two cells? Well, it turns out the strategy in animal cells is different compared to what plant cells do. Let's see what strategy each type of cell employs. As we've seen before, animal cells are kind of like water balloons, and the cytokinesis strategy employed by animal cells is reminiscent of this fact. As mitosis is ending, cytokinesis begins to divide the forming nuclei and the rest of the contents of the cell into two new daughter cells. This basically looks like you looped a string around the outside of a water balloon and started to constrict it like a drawstring. Now, obviously, the real-life outcome of this example doesn't really end too well for us, since we end up with a face full of water. Luckily for the cell, though, the outcome is much less messy. During cytokinesis, the role of the string is played by the contractile ring. Now, a contractile ring is composed of actin filaments that are assembled just below the cell membrane. A protein called myosin works with the actin filaments to constrict the ring till the cell membrane touches in the middle. Now, once those membranes pinch together, they fuse, and the result is two new cells, each with one nucleus, complements of mitosis, and half of the cytoplasmic content, complements of cytokinesis. So why do plants utilize a different cytokinetic strategy? Well, it's not just because they're trying to be different. Remember that plants have a cell wall. Now, since that cell wall won't bend, a contractile ring isn't going to work. But nature's nothing if not creative. Remember that the Golgi apparatus is an organelle that plays a major role in secretion of molecules from the cell. Now, it accomplishes this by releasing tiny membrane sacs called vesicles. And a vesicle containing a molecule destined for secretion buds off of the Golgi apparatus and heads for the cell membrane. Since it's made of phospholipids, just like the cell membrane, a vesicle can fuse with the membrane. And in this way, the molecule is released. Now, this reveals a strategy that the plant cell can employ to form a new cell membrane without a contractile ring. In a plant cell, a new membrane is formed by vesicles, which fuse together in the middle of the cell to form what's called a cell plate. Now, as the cell plate expands to the edges of the cell, it can fuse with the cell membrane and form two new cells in the process. Okay, so we've solved one problem, right? But we've just created another. Now we have two cells, but they're stuck in the same cell wall enclosure. Well, remember how we said that the Golgi apparatus is good for transporting molecules outside the cell? Well, what if we pack some cell wall ingredients in the vesicles that we use to form the cell plate? So if we do that, once the cell membrane is formed, these secreted molecules can assemble into a new cell wall, and in this way, the two plant daughter cells can separate. In summary, Cytokinesis is the process in which the cytoplasmic content of the cell is divided into two new daughter cells. The mechanism in which cytokinesis is achieved differs between animal and plant cells. A contractile ring is a ring of actin filaments located below the cell membrane, which constricts the middle of a dividing animal cell to complete cell division. The cell plate is the area where the new cell membrane and cell wall form between dividing plant cells during cytokinesis.